A coarse-grained protein from a bond-fluctuating polymer, fake computer simulation. Bond fluctuation model for polymer chain was introduced by Karmazin and Kramer in 1988 with a computer simulation of a polymer chain on a square lattice. Since then, it has been extended to cubic lattice and has been extensively used to address a number of complex problems for macromolecular systems. Following are two original references. 1. I. Karmazin and K. Kramer, Macromolecules Volume 21, page 2819, 1988. 2. Monte Carlo and Molecular Dynamics Simulations in Polymer Science edited by K. Binder, Oxford University Press, 1995, Chapter 1 and 6, it will be referred as MCMD binder. A polymer chain is a set of monomers, i.e. nodes, held together with covalent bonds. Shape and size of a monomer can vary depending on the degree of specificity and approximate representation. Generally, a set of atoms and molecular units can represent a node of a polymer chain. The illustrative diagram from MCMD binder and a model snapshot cover the basic idea behind representing a polymer by a chain. Structure and dynamics of constitutive elements are correlated as the global properties result from their cooperative and competing mechanism. Before examining the stochastic movement of a polymer chain, it would be good to look at the stochastic movement, random walk, of a free monomer/particle first. The trajectory of a random walk over time is an illustration of the ideal chain, Flory. Variation of the average mean square displacement with the time step describes the dynamics. A linear dependence of the mean square displacement on time shows that the dynamics is diffusive. What happens when many particles/monomers are tied together in a polymer chain? A monomer cannot share the same space with the others at the same time, known as excluded volume effect or hardcore interaction. The random movements of each monomer are constrained by bonding to adjacent monomers. Obviously the dynamics of monomers is expected to be different from that of a random walk at least at short times. The dynamics is slow in a short time before it becomes diffusive. In fact, the short time dynamics is a well-known Rouse dynamics where the root mean square displacement of the monomer depends on time step with a power of 1/4. A crossover occurs from Rouse dynamics at short time where the root mean square displacement varies with time to a power of 1/4 to diffusive at long time where the root mean square displacement varies with time to a power of half. In a crowded polymer melt, the dynamics becomes even more complex, slithering snake reptation model of Degenn and tube model of Doi Edwards. Each monomer exhibits a well-known multi-scale dynamics, Roused reptation to post-reptation to diffusion where the root mean square displacement of the monomer depends on time step with a power of 1/4, 1/8, 1/4, half. With traditional molecular dynamics and Monte Carlo simulations in a continuum space, one can hardly reach from Roused to reptation due to huge degrees of freedom in a complex melt which requires a relatively high concentration of large polymer chains in a simulation box. Coarse-grain representation of constituents and underlying matrix is a viable approach along with other tools and tricks to probe large-scale properties of such systems. Bond fluctuation mechanism is an effective and efficient coarse-grained approach with the ample degrees of freedom with the flexibility to enhance with fine graining in a discrete lattice space. On a square lattice, a monomer is represented by a square unit cell. If available, it can hop to one of its 8 adjacent sites, i.e., 0, 2, 1, 2, 8, by 1 and square root of 2. Lattice spacing. There are 6 different bond lengths, 2, square root of 5, square root of 8, 3, square root of 10, and square root of 13, in unit of lattice constant. 36 bond vectors and 41 angles between adjacent bonds. On a cubic lattice, a monomer occupies a unit cubic cell, with 8 lattice sites. A free monomer can hop to one of its 26 adjacent cubes if available. There are 108 bonds with 5 different bond lengths, 2, square root of 5, square root of 6, 3, and square root of 10, in unit of lattice constant and 87 different angles between adjacent bonds. A bond length square root of 8 is generally excluded to avoid bond crossing and maintain simplicity. Stochastic movements of a bond fluctuating polymer chains. Stochastic movements of a bond-fluctuating polymer chains. Proteins are heteropolymers of 20 amino acids. Specificity of amino acids, residues, and its sequence in a protein is critical for its unique function. Broadly speaking, based on hydropathy index, 8 amino acid are hydrophobic, 8 are polar, and 4 are electrostatics. A protein such as alpha-synuclein consists of 140 amino acids in a unique sequence with a diverse range of hydropathy index. Specificity of amino acids, their number, and sequence in a protein is critical for its unique functionality and versatility. Ability to organize into specific morphology and its response to such parameters as temperature, field, solvent quality, concentration, etc. makes it one of the most interesting topic of investigation. Despite enormous efforts for decades, how proteins fold remains an open question. All relevant approaches involving atomistic scale details to coarse grain and multi-grain descriptions with various tools such as Monte Carlo, molecular dynamics, and its variants are highly desirable to identify specific and sub-universal characteristics of proteins. A coarse-grain representation of residues with bond-fluctuating mechanism on a cubic lattice with ample degrees of freedom to investigate protein is an alternative and supplementary method along with current standard tools. A knowledge-based residue-residue interaction matrix with 210 independent pairs among 20 amino acids is a good choice to begin with. The knowledge-based residue-residue interaction has been developed over decades from a huge ensemble of protein structures in Protein Data Bank and is improving with expansion of a rapidly growing list in PDB. A generalized Lennard-Jones potential between residues with the input of knowledge-based residue-residue interaction can be used as phenomenological interaction to carry out Monte Carlo simulations. Local and global physical quantities such as contact map, energy and mobility profiles, mean square displacement of the protein, radius of gyration, and its structure factor etc. can be analyzed. Stochastic motion of alpha-synuclein protein. A contact map, neighboring residues along the contour within the range of interaction, of protein configurations at the end of 10 million time steps with 40 independent samples at different temperatures shows how the thermal noise reduces the degree of contacts. From variations of the structure factor SQ with the wave vector Q1 can quantify the degree of compactness, via effective dimension, or globularity of the protein for a range of temperature. Following conclusions are drawn from a comprehensive analysis of simulation data of coarse-grain model of alpha-synuclein. 1. Based on mobility profile 3 segments are identified, N-terminal, residue 1 to 60, and C-terminal, residue 96 to 140, separated by the high mobile non-amyloid component, NAC, residue 61 to 95. This is already known from other analysis, so what is the big deal? Well, this verifies the reliability of the coarse-grain approach. 2. The intraprotein contact profile shows a higher frequency of residue aggregation, clumping, in the N-terminal region relative to that in the C-terminal region, with little or no aggregation in the NAC region. This was not as clear as it is observed from this analysis. 3. The radius of gyration of alpha-synuclein decays monotonically with decreasing temperature consistent with available results. 4. Estimates of an effective dimension, D, show globular structure at low temperature with D about 3, a random coil at high temperature with D about 2, and in between 2 and 3 at the intermediate temperatures. Nowhere else, the higher order structures of proteins are quantified as here with an effective dimension. Advantage: because of simplicity of the lattice structure, it is easier to develop flexible and efficient computer codes for specific problems. Response of thermodynamic quantities to temperature, concentration, and nature of the underlying binding substrate and solvent interaction is the main focus. More complex problems can be analyzed with large-scale simulations. Disadvantage: making a quantitative comparison of the physical quantities with a laboratory measurement at absolute values of parameters is not feasible with such coarse-grain approach. Instead, one can identify the trends and variations with respect to changes in magnitude of parameters. Models are too simple to represent real laboratory samples. Potential, development and extension, already done, planned, or in process. Fine graining, an amino acid can be represented by 2, 3, or more nodes. B. One can use 4 nodes to incorporate some specificity of C-alpha backbone, C and N terminals, and the psi group, R. More refined force fields can be developed and used as input. Parameters and reduced units can be calibrated with atomic scale MDN or laboratory data.